So now we're going to switch gears and talk about eukaryotic cells. So in the previous section, we learned about how simple prokaryotic cells are. There's not a lot of structure there. It's just loosely floating substances inside of this storage container, really. And they have some DNA, they have some ribosomes, which are really just folded pieces of RNA, and some protein. And that's basically it. They've got some external structures. Their shape really defines what they are. Eukaryotic cells are much more complicated. So we're going to start by talking a little bit about an overview of animal cells and plant cells, and then I'm going to talk about the cell membrane structure. First of all, plant cells and how plant cells are different from animal cells. Plant cells have a cell wall, and that cell wall is going to give the plant cell a rectangular or square shape. So it kind of fixes the shape for the plant cell. It allows these cells to stack in rows. Now the term cell, if you think back of where did the term cell come from? So Robert Hooke was this guy, way back when, that decided to take a thin slice of cork material, and cork comes from a plant. So then he looked at it under using a magnifying glass, a very powerful magnifying glass. And he noticed that the entire structure looked like rectangles. And those rectangles to him looked like these things called cellula that the monks used to go into or something like that. So way back when, he made this association that, hey, they look like the cellula of monks. So he coined the term cell. He said, these are what cells are. So this cell wall is very important in history. It gives this shape to the plant cell, and it's also kind of where cells got its term from that shape. An animal cell does not have a cell wall. Animal cells are our cells. We are animals, so they do not have a cell wall. Other eukaryotic organisms like fungi and protozoa and algae, they vary in whether they have a cell wall or not, but animal cells do not have a cell wall. All of these organelles that you see listed here, we're going to go through those in the next section. But this section, I just want to talk about the membrane. So a membrane surrounds each cell, no matter whether it's prokaryotic, eukaryotic, if it's multicellular, unicellular organism, you're going to have a membrane. That membrane defines the boundary of the cell. And every membrane in every living organism is made up of phospholipids, and it's a phospholipid bilayer. Now some organisms have an inner membrane that's the phospholipid bilayer, and then an outer membrane that's part phospholipids and part something else. So those organisms, though, still have at least one membrane that surrounds them that's made up of a phospholipid bilayer. So just a review of what the phospholipid is from last week's lecture. A phospholipid has this phosphate head that is hydrophilic or water-loving, and lipid tails that are hydrophobic or water-fearing. So the hydrophilic heads are on the outermost surfaces, while the hydrophobic tails kind of line up and join each other in the center. Because of their chemical structure, phospholipids spontaneously form bilayers in water. So if you have a whole bunch of phospholipids and you put them in water, they're going to spontaneously come together to form a membrane-like structure. All of the lipid tails are going to be on the inside, while the phosphate heads are going to be on the outer sides of the membrane. This membrane is selectively permeable. The only substances that can pass through are going to be lipids and small nonpolar molecules. So what selectively permeable means is that it only allows certain substances to pass through. It's selectively So it chooses which substances can go through, can permeate or pass through the cell. Ions and large polar or uncharged or charged molecules, they require special channels in order to get through the membrane, and we'll learn more about all of those transport mechanisms in next week's lecture after the first exam. Membranes are much more complicated than what they kind of seem. So in a cell membrane, we have all of these proteins. Some of the proteins pass all the way through, some of them are just on the surface, and then protruding out of the surface we have this glycocalyx layer. We have all these different types of sugars, proteins, and lipids and other types of molecules that are going to serve as receptors or enzymes or do different functions on the surface. So now we'll go through a list of some of the things that you'll find inside of a membrane. First of all, cholesterol. And we learned about cholesterol in last week's lecture, but here you have a molecule of cholesterol. Cholesterol has 4 rings, and it wedges itself right in the membrane. If you have too much cholesterol, this cell membrane becomes very rigid, which makes it prone to lysis or rupturing. If you don't have enough cholesterol, these phospholipids can slowly start to float away and become really loose, and the cell becomes really leaky, and then it loses its selective permeability, and toxins and stuff can start to seep through the membrane, and it becomes a very dangerous situation for the cells. So we need some cholesterol, but we don't want too much. The next thing you'll see is transport proteins. So transport proteins transport substances through the membrane. Enzymes are going to perform chemical reactions on the surface of the cell. Recognition proteins. Recognition proteins are known as antigens. And I like to call antigens cell ID cards. So if you want to get into a bar, you got to show your ID and show that you're 21, and then you're allowed to enter. If you want to rent a library book, you may have to show your library card, and then they'll let you check out those books. So on the surface of every cell, there's a series of specific molecules known as antigens, and those antigens give that cell its identity. Now in your body, you have an immune system. You have all these white blood cells, and it's their job to float around all over your body, bump into cells, and decide, is this a good guy or a bad guy? Does this belong in the body, or does it not belong in the body? Now these white blood cells don't have tiny eyeballs. They're dependent on the interaction between these antigens and their receptors. So your white blood cell bumps into the surface of the cell, hits these antigens, and has to make a decision if these antigens look normal or abnormal. If they look normal, they'll leave it alone. But if they look abnormal, then your white blood cell knows to attack and kill this cell. So antigens or recognition proteins, they play a major role in giving that cell its identity, but also in determining what is self and what is not self, so that your immune system attacks foreign things and leaves your own cells alone. Then we have adhesion proteins. Adhesion proteins we go through in cellular junctions in the very last couple slides in this PowerPoint series. So in this week's lecture, we will talk about the different types of adhesion proteins. But what these do is they allow cells to stick together to form tissues. And then we have receptor proteins. So receptor proteins are going to pick up on signals from the environment or from neighboring cells. So receptor proteins are going to interact with chemicals, they can interact with hormones, neurotransmitters, and even just temperature changes or pain sensations. They have the ability to pick up on all sorts of different things. There's certain receptor proteins called the photoreceptors in your eye, and those pick up on light and changes of light intensity. So these receptor proteins pick up on signals. The cell membrane is described as being a fluid mosaic. What this means is if you were to take a picture of the surface of a cell and then take the same picture in the exact same spot minutes later, you would be looking at two different pictures. Because these proteins are constantly moving. So it's not like this protein stays there forever. This protein is floating around all the time. So these are constantly changing their association in the membrane. It's not stagnant, it's constantly changing. Fluid, if something's fluid, it's movable. Mosaic, if you've ever seen a mosaic before or made a mosaic, it's a bunch of little pieces that come together to form a bigger picture. So that's kind of where that came from. But if you ever hear the term fluid mosaic or the fluid mosaic model, it's just talking about how stuff in the membrane moves around and shifts so that it's constantly in motion.